Good morning, everyone. It's a great pleasure to be here today and share with you my research done at the University of Naples in Italy that is mainly based on preclinical studies on N-acylethanolamines modulation of pain transmission and the related maladaptive neuroplasticity. Only a few words on the pain transmission. So, nociceptive pain occurs when nociceptive system is activated by noxious stimuli, and it is divided in 5 different phases: transduction, conduction, transmission, perception, and modulation. Nociceptive pain originates into the nociceptors that are highly specialized primary sensory neurons that transform the noxious stimulus into an electrical signal that is conducted along the axon to the cell bodies into the dorsal root ganglia and then to the central terminal into the spinal cord. Here, the electrical signal stimulates the release of neurotransmitters, including glutamate and substance P, that activate the receptors, their receptors, and induce excitatory postsynaptic responses. These excitatory inputs is sent through the ascending nociceptive pathway to the higher center in the brain, where it is perceived as pain. But on the other side, pain is modulated by inhibitory descending projection into the dorsal horn of the spinal cord, and this inhibition serves as to provide analgesia at time of danger. So pain does not compromise normal functions. But when we talk about neuropathic pain, this is something different from nociceptive pain because this is a chronic pain and it is induced by arthritis, peripheral nerve injury, for instance, cancer, chemotherapy treatment or cancer, and it is characterized by neuroinflammation. Neuroinflammation is driven by glial cell activation into the spinal cord and the infiltration of immune cell into the central nervous system, and they release pro-inflammatory mediators and gliotransmitters that remodulate synaptic plasticity and induce central sensitization. Central sensitization is the main neural mechanism at the basis of development and maintenance of chronic pain that is characterized by alteration of pain perception, and the two main symptoms are called mechanical allodynia and thermal hyperalgesia. But it's known that chronic pain induces morphological modification into the different areas in the brain, including hippocampus, periaqueductal gray, amygdala or medial prefrontal cortex. And this brain plasticity and the synaptic reorganization is responsible for the comorbidities of chronic pain like mood and anxiety disorders or cognitive decline and memory deficits. In particular, when after a peripheral injury, a serial of rearrangement happen into the different nuclei of amygdala that control nocifensive responses and emotional and negative affective states like anxiety. Moreover, a few years ago, Apkarian and colleagues demonstrated that patients affected by different types of chronic pain, they showed a reduced hippocampus volume, both left and right, as compared to healthy volunteers. Moreover, in vivo studies show that also neuropathic rats have an altered synaptic plasticity into the CA3 in the hippocampus, together with the reduced neurogenesis into the dental gyrus in the hippocampus. Actually, central sensitization, it's due to an altered glutamatergic signaling and loss of tonic inhibitory control that induce spinal neuronal wind-up and long-term plasticity with consequent amplification of pain transmission to the brain. However, not many years ago, researchers realized that besides GABA and glutamate, also endocannabinoids are key modulators of synaptic transmission into the brain, into the central nervous system. And as we know, endocannabinoids, through their retrograde action mechanism, they activate the presynaptic receptors by modulating tonic inhibition and long-term depression. But when we talk about endocannabinoids, we only mention a class of lipids belonging to a wide family of endogenous fatty acid amides, recently defined as an endocannabinoidome. And as already said by the previous speakers, the two most studied endocannabinoids in the brain are the 2-AG and the anandamide. They are synthesized in the brain after the calcium influx into the postsynaptic neuron and the activation of NAPE-PLD enzyme that synthesize anandamide and palmitoylethanolamide in 1:9 ratio, whereas the synthesis of 2-AG is mainly dependent on the activation of DAG lipase alpha enzyme. Action mechanism, we have seen that of endocannabinoids, we have seen that are different. So they activate transmembrane receptor, CB3P1 or the orphan receptor, but they also interfere with the activity of enzyme of degradation by enhancing endocannabinoid levels, and some of them are agonists of the nuclear receptor PPAR-alpha. Cannabinoid receptors are widely spread into the whole body, but they are highly localized into the brain and also expressed in the peripheral organs and tissues where they control a lot of physiological function. In particular, CB1 receptor, G inhibitory coupled receptor, higher expressed into the hypothalamus and limbic system, negatively modulate feelings and emotions, whereas its expression in the peripheral tissues mainly mediate metabolic effects. CB2 receptor expression instead, it has been mainly found on peripheral immune cells, including macrophages, microglia, B cell, and T cell, and it has been, in particular, it has been found, like further investigation on the pattern expression of CB2 have revealed that CB2 is also expressed in the brain, but differently from CB1, it's present on microglia cell, and it has been hypothesized that the activation of CB2 receptor mainly mediate the anti-inflammatory and neuroprotective state of microglia. So nowadays the monocentric neuronal view is shifted toward a larger view of that include an orchestra of different cells that playing together modulate synaptic plasticity in the brain, and these cells include microglia, astrocytes, oligodendrocytes, and also peripheral immune cells. In particular, microglia cells represent the resident immune cell in the brain, and in physiological condition, it is it presents a ramified morphology with long processes and a small soma, and we call it resting microglia. Resting microglia are not sleeping cells, but they are in charge to patrol the brain environment. But when something happens, like a brain damage or an inflammatory insult, microglia change its morphology, so it retracts the processes, enlarge the brain, and so we call it activated. That it is able in this way to clean the brain from the cell debris and dysfunctional synapses. There is an in vivo study that demonstrates the key role of CB2 receptor activation in nociceptive responses in neuropathic pain. Indeed, they show that the induction of neuropathic pain in CB2 knockout mice induces a more severe symptoms of neuropathic pain like mechanical allodynia and thermal hyperalgesia. And there are also other studies that demonstrate that the activation of CB2 receptor mediates the analgesic effects of the palmitoyl ethanolamide in chronic pain models. So palmitoyl ethanolamide, we have seen that it's an endogenous fatty acid amide, but it is also found in foods like peanuts, milk, or egg yolk. And it has been discovered by Rita Levi-Montalcini, who received the Nobel Prize in the 1960s. And this is hydrolyzed in the brain by the fatty acid amide hydrolase, whereas in the macrophages by the NAAA enzyme. And palmitoyl ethanolamide is the endogenous agonist of PPAR-alpha receptor, that it's a nuclear receptor that in the cytoplasm dimerize with the receptor of retinoic acids, translocate to the nucleus and binds to the PPAR responsive elements on the DNA by modulating expression of different genes. But on the other side, PIA is also defined as endocannabinoid-like compound because it induces an increase of anandamide levels indirectly by interfering with the Fa enzyme activity. But recently it has been hypothesized that PPAR-alpha activation, that the PIA is able to induce the expression of CB2 receptor. Indeed, in our research group a couple of years ago, we have published a paper where we have shown for the first time that palmitoyl ethanolamide, through its genomic action mechanism, is able to induce the CB2 receptor expression that is involved into the microglia morphological and functional changes. In particular, so this is an in vitro study. We performed it, we cultured the microglia cell from the neonatal mice, and what we observed was that the application of the highest concentration of PIA induced an increased expression of mRNA and protein level of CB2 receptor, and this effect in the protein, on the protein, was blocked by the PPAR-alpha receptor antagonist. This result was also confirmed by immunofluorescence analysis, where we also found increased CB2 receptor expression in PIA-treated cells as compared to vehicle-treated cells, and the same was observed in cells treated with the PPAR-alpha agonist and with the positive control interferon gamma. Moreover, we found that the same treatment induced an activation of microglia. Indeed, we can see an amoeboid shape, so it's an activated microglia as compared to vehicle-treated cell, where we can still see long processes and the small soma. At this point, to demonstrate that this effect was mediated by PPAR-alpha receptor activation, we silenced the mRNA expression of CB2 in the microglia cell. And we found that silent cells that had 80% less of expression of PPAR-alpha, they did not show any change in CB2 receptor expression after treatment with PIA, differently from non-silent cells, where the treatment with PIA or with the other agonists, they increase significantly the expression of CB2 as compared to non-treated cells. And bioinformatic and biochemical analysis demonstrated that the complex, that the one cell stimulated with PIA, the complex exactly bound to two consensus sequences on the DNA that were found in the CNR2 gene. One more thing was that we, in another set of experiment, we infected microglia with bacteria, Porphyromonas gingivalis, and we found that PIA increased the phagocytosis and intracellular killing, so confirming it's also another effect that is an antibacterial effect. Moreover, treatment with PIA also increased the migration of cells toward the 2AG source as well as the PPAR-alpha agonist, and this effect was, as previously in the other experiment, was blocked by PPAR-alpha and CB2 receptor antagonists. Moreover, treatment with PIA also increased the So, based on the evidence of anti-inflammatory and analgesic effect of PIA in a different pain model, we performed also in vivo studies in an animal, in a mouse model of neuropathic pain, where we induced neuropathic pain through the cut and the partial cut and the ligation of the sciatic nerve. And 2 weeks after the surgery, we started treating daily the animals with PIA or vehicle for 2 weeks. And so at the end of treatment, 1 month after the injury, we started our experimental evaluations. So first, we found that SNI animals, they developed mechanical allodynia. So it means that when we touch the paw of animal with a very thin filament, like an hair, they retracted the paw. So they felt pain differently from the non-injured animal. And the chronic treatment with PIA significantly reverted this symptom. But intriguingly, we found, as observed in humans, that also neuropathic mice developed some comorbidities like alteration of working memory, discrimination memory, and also they showed a depressive-like behavior measured by the increased immobility time into the tile suspension test. And all these behaviors were reverted by chronic treatment with PIA. These animals also showed anxiety-like behavior, but PIA on this task failed to work. The depressive-like behavior and all the cognitive alteration indicates the involvement of cortical areas that usually control high cognitive functions. Indeed, from in vivo extracellular recording, so we recorded the activity, neuronal activity of neurons in the medial prefrontal cortex in anesthetized animal by the application of a noxious stimulus on the paw, and we found an overexcitability of neurons into the neuropathic animals 30 days, but even more 2 weeks after injury, as compared to to control animals that we call sham animals. Moreover, it's known that depression, so negative affective states in general, they are linked to alteration in neurotrophic factors like BDNF. Indeed, we found that SNA animals had a decreased level of BDNF and increased level of the receptor TrkB, probably as a compensatory mechanism, and the chronic treatment with PIA reverted these modifications. Indeed, antidepressant drug, we know that the strategies to increase BDNF release, like for instance ketamine or scopolamine, that by blocking an MDA on GABA interneuron, this inhibit the release of glutamate that increase the intracellular calcium and induce BDNF release. BDNF in turn activates TrkB receptor that induces the activity of the mammalian target of rapamycin-1 that is involved in increased synaptogenesis, underlying the rapid antidepressant effects. But mTOR complex is, by recruiting different protein and mediators, is involved in plenty of functions like cell survival, cell growth, or inhibition of autophagy. Already in the 18th century, a German philosopher, Immanuel Kant, in its theory a priori formen, he defined the cognition as something that begins with experience, but it is not caused by experience. And he also defined time and space. So in some way, he predicted the existence of some brain structure that are able to control cognition, learning, and memory. And this is hippocampus that many years later has been discovered. And on a hippocampus, we performed another in vivo study where in neuropathic mice, both wild type and PPAR alpha knockout. And of course, we confirmed that these animals developed mechanical allodynia and thermal hyperalgesia, but the chronic treatment with PIA reverted these two symptoms in wild type but not in knockout mice. And one more time, we found in these animals an reduced recognition memory and also an altered spatial memory, because animals spend less time into the target quadrant, into the Morris water maze test. So we usually put a platform under the water in a swimming pool. They learn for 5 days where the platform is located. After 5 days, we remove it. And so we see if they remember where the platform was located. And we can see from the track plot recorded by the camera that SNAI animal, they just went around and around because they did not remember. And the chronic treatment with PIA reverted this alteration only in wild type but not in knockout mice. Functionally, the neural mechanism at the basis of learning and memory is long-term potentiation. So this is what happens when we try to learn and remember something. So it's a mechanism that I simulated in anesthetized animal by applying electrical stimulation into the lateral entorhinal cortex and recording the excitatory postsynaptic field responses into the dentate gyrus. So I analyzed the pathway. And if we look at recording, this is an example of a recording in a normal animal. So after we apply single pulse, and we get a basal response into the dentate gyrus. But when we apply a tetanization, so a theta bar simulation, we have an increase of the magnitude of the responses. So it means that the person, the animal, can learn and remember, can develop long-term memory. Indeed, if we look across the bar graphs, we can see that the recording in normal animals, so the application of the tetanus induced an increase in the magnitude of postsynaptic responses that was from 1 to 3-fold higher than the baseline, whereas the application of the same protocol in SNAI animals that already showed a higher basal synaptic responses did not induce LTP, because we did not observe changes in the parameters like amplitude and slope. And the chronic treatment with PIA partially restored this alteration in wild-type animals, but surprisingly, we also in PPAR alpha knockout mice, we still could see some LTP, hypothesizing other action mechanism of PIA. These behavioral and functional alterations were associated with the glutamatergic signaling modifications. Indeed, we found reduced number of asymmetric synapses in neuropathic mice, increased extracellular level of glutamate into the dentate gyrus, and increased expression of AMPA receptor subunits. And these modifications were all normalized by chronic treatment with PIA. A few years ago, it has been demonstrated that chronic treatment is induces morphological modification in the medial prefrontal cortex. Indeed, Metz and colleagues showed that they found in neuropathic rats a higher arborization of basal dendrites of neurons into the medial prefrontal cortex as compared to control animals. And as they found in medial prefrontal cortex, also in the dentate gyrus, in the hippocampus, we found an important reduction in dendrite arborization into the neuropathic rats as compared to control animals. Also, we found a reduced volume of dentate gyrus and a reduced neurogenesis, measured by the reduced number of newborn neurons and double courting positive cells. So finally, we, as already observed in vitro, in other studies, we confirmed that this pathway that I analyzed, lateral entorhinal cortex dentate gyrus, is mediated by endocannabinoids, because when I microinjected the CB1 receptor antagonist into the lateral entorhinal cortex, I got a decay of synaptic response, and I didn't see any LTP, any potentiation. And also, when we measure the levels of endocannabinoids into the lateral entorhinal cortex in SI animals, we found an increase in 2Ag level, significantly both contra and ipsilateral to the lesion, that was associated with the CB1 receptor colocalization with beta-arrestin. So it means that CB1 internalized, probably to compensate the high level of 2Ag into the cortex. So, in conclusion, we can say that one more time we have confirmed that chronic pain induces nociceptive alteration in nociceptive responses together with central comorbidities like anxiety, depression, and memory deficits. And the chronic treatment with PIA ameliorated all the behavioral alteration, reverted the LTP, the impairment of LTP, and also recovered all the altered glutamatergic signaling. So we think that cannabinoid-related molecules might represent a pharmacological tool, a promising pharmacological tool to treat chronic pain and all the associated negative affective disorders. So I would like to thank my research group in Naples, all our collaborations, and of course all of you for the attention.
On a le temps pour une ou deux questions.
Oui, on peut acheter du PEA comme complément alimentaire, mais donc on le prend sans suivi médical, etc. Que pensez-vous du fait actuellement pour quelqu'un d'utiliser le PEA, notamment pour traiter des douleurs chroniques chez l'homme actuellement?
I missed the first part of the question, but I understood the sense. So you asked the use of PIA in patients for the treatment of chronic pain. Yeah. So PIA, it's already available as a dietary supplement, and it is so used for the treatment of inflammation and different types of chronic pain. And it is so it's used for several types of pain associated with multiple sclerosis, orofacial pain. Did you want to ask something else? No?
Que pensez-vous de l'utilisation du PEA actuellement chez l'homme? Est-ce que c'est positif? Est-ce qu'il y a des effets?
Yeah. Okay. So the advantage.
The use in men today, is it positive?
Yeah. It is. It has different characteristics that makes it an attractive drug for pain relief, because it does not. We have seen that it does not bind directly to the CB receptors. So it over the time, if you use it for a long time at very high dosage, it doesn't give side effects, doesn't give like tolerance, dependence, neither drug-drug interaction. Indeed, today, as in medical use, it is prescribed in association with the traditional anti-inflammatory drugs, non-steroidal and steroidal drugs. So in this way, you can reduce the dosage of the anti-inflammatory drugs and reduce the side effect.
Oui, bonjour. Merci pour votre présentation. Moi, je suis médecin de la douleur. Je travaille avec des gens qui ont des douleurs chroniques depuis des années et des années. Ma question, c'est, voilà, enfin, c'est très intéressant cette étude. Ça ouvre des perspectives pour mes patients, justement. Mais dans votre étude, vous présentez les souris, en fait. Vous avez fait des tests sur les souris pour le moment, c'est ça? J'ai bien compris. Il n'y a jamais eu d'études chez l'homme. Et les souris, quand vous testez la douleur qu'elles ont, elle dure depuis combien de temps, en fait? Quand vous testez les souris, depuis combien de temps elles ont cette douleur chronique. Parce que moi, quand les gens viennent me voir, c'est au bout de 5 ou 10 ans de douleur chronique en général. Donc les remodelages au niveau cérébraux sont quand même plus complexes. Je pense qu'il y a les interactions avec la cognition, avec la mémorisation de la douleur. Il y a des interactions aussi avec des changements de comportement, notamment une kinésiophobie. On voit des gens qui ne sortent plus de chez eux. Il y a toute une intrication avec d'autres choses, en fait, bien plus complexes. Et voilà, c'était pour voir un petit peu si l'application chez l'homme, on pouvait déjà dire que ça pouvait être intéressant, ou bien s'il fallait peut-être quand même déjà tester sur des patients, on va dire, qui ont des douleurs depuis plus longtemps, quoi. C'est un peu...
Thank you for your question. This is a good point because there are a lot of evidence of preclinical studies where PIA works very well. So it's super fine. But the problem is that when we move to patients, because we don't have experience with the patients, but when we talk to doctors who prescribe PIA, they sometimes complain that after a few months, so they get the maximum effect, but then the effect starts disappearing. This is the problem that it's hard to fix because over the time there is like an adaptation of all the molecular systems. So the receptors, as you can see, when at this point, like the endocannabinoid levels of some endogenous compound increase, so the receptors they internalize, so you have a desensitization.
Same effect with opioids or
yeah, it's
always the same.
Yeah, it's the same.
It's not like acute pain.
No.
It's not the same mechanisms, and we know that there's a lot of cognitive effects. Yes.
Yeah, this model that we applied, so the spare nerve injury, we used it because it's a model of long-lasting pain. So if you cut the nerve and tight it after 1 year, we also are studying old neuropathic animals. So after 1 year, they still have pain, and so allodynia, paralgesia, and they have some memory and cognitive dysfunction. Yeah, but I agree with you that this is
It's not miraculous.
It helps.
At the moment, perhaps.
What we claim about this PIA as compared to the other compound, cannabinoid-like compound, is that it doesn't give. You can take it until until 1.2 grams per day for a long time, and they don't complain about side effects differently from others' drug, and they don't interact with others' drug. So the strategy might be to combine PIA with the other endocannabinoids.
Perhaps at the beginning to induce a movement or a changement in the patient, and then
yeah.
Oui, voilà, c'est. Je suis pas très bonne en anglais.
And then switch back. Okay, thank you very much.
Thank you.